Hello students, today is the second online bio class for class 10. Now we were doing genetics. We have done up to monohybrid cross last day, but I'll show you the other part of the monohybrid cross again today. Now today I'm starting with dihybrid cross. Now by di means two. When we are taking two characters at a time and crossing with each other right from the parental generation, then we are getting the required phenotypic ratio. Genotypic ratio for dihybrid cross is not there in your syllabus, so you don't have to do it. Now when we are starting with parental generation, here we are showing a cross between the round yellow seeds of a pea plant. With that we are crossing wrinkled green seeds of a pea plant. Now as you can see, we have taken two characters at a time. One is the shape of the seed, another is the color of the seed. Now two opposite things we have taken together in order to cross. And in the F1 generation we are getting, it's very easy, we will take one gene at a time. Capital R, small r, capital Y, small y. So this is the genotypic or the genotype rather, which we are getting in F1 generation, and then we are self-crossing this genotype with the other genotype. Okay, so we are crossing one capital R, capital Y, small y with another capital R, capital Y, small y. Now in F2 generation, we are doing again the Punnett square or the checkerboard. Here again we have drawn the squares, 4 squares in each of the axis, and again we have given male and female sign, and in the boxes over the squares we will take the probable genetic combinations at a time, like capital R, small y, capital R, capital Y, small r, capital Y, small r, small y. Now the sequence which we have written in any one of the axis, the same sequence we will maintain on the other axis. It may be any one of the axis, and then again we will simply multiply it with one another and we'll get such combinations as I have shown here. Now from after getting the combination, we have to take out the phenotypic ratio from here, because genotypic ratio is not there in your ICSE syllabus, so we'll only focus on phenotypic ratio. Now when we will find out the phenotypes, the similar phenotypes, then what we'll do, we will give a sign so that we can understand while calculating. Here I have given a very simple sign, that is tick mark. You can see that the similar genotypes I have ticked with single tick. Okay, capital R, capital Y, capital R, capital Y, we have taken 1, 2, 3, 4, 5, 6, 7, 8, 9. You can see there are 9 single ticks. Please see properly. We have got capital R, capital Y genetic constitution in 9 alleles or allelic combinations. Okay, so there are 9 round yellow seed pea plants we are getting in the offspring. Clear? So the first ratio is 9. Then we will look after the second kind of phenotype, that is round green seeds, that is capital R, small y. We have got I have given 2 ticks for that. You see 1, 2 and 3. There are 2 ticks which I have given, capital R, small y. I hope it's clear. So we have got 3 round green seeds pea plant. Next we will see the third kind of phenotype, that is wrinkled yellow seeds. That means small r, capital Y. Now we have got 3 ticks. You can see in the Punnett square 3 ticks. Small r, capital Y. So small r, capital Y, we have got 3. So again, next ratio is 3, and only the left out is wrinkled green, that is small r, small y, that is only 1. So our required phenotypic ratio is 9:3:3:1. I hope it's clear. It's given in your book also. Please go through your textbook. Now, last day we have done a monohybrid cross class. Now, today I am showing you that if one of the parents is not homozygous dominant but heterozygous dominant tall pea plant, then what phenotypic and genotypic ratio we will get. Last day I have shown you that there are two capital T's. That means homozygous dominant pea plant I have crossed with the homozygous recessive dwarf pea plant. Now today I have taken one heterozygous dominant tall pea plant and I am crossing with homozygous recessive dwarf pea plant, and I am getting here 2 capital T small t and 2 small t small t after crossing with each other. And in the F1 generation, I am getting, when I am taking common from these 4 genotypes, I am taking capital T small t and crossing with small t, the same genotypes which we have got in the parental generation. So it's very easy. Now in the F2 generation, again when we are drawing the checkerboard, we are segregating the genes similar way, and we are getting the phenotypic and genotypic ratio similar here, that is 2:2. That means coming 1:1, because capital T small t means tall pea plant, and small t means dwarf pea plant. So we are getting 2 tall pea plants and 2 dwarf pea plants. That means 1:1 ratio. Okay. Similarly, when we are going through the genotypic ratio, in the genotypes we are seeing again similar. Capital T small t there are 2, small t there are 2. That means genotypes also same ratio, 1:1. Next, another very important part is sex-linked inheritance. It is the appearance of a trait which is due to the presence of an allele on either the X chromosome or on the Y chromosome. This is a mode of inheritance which can be found in human beings because of an allele which is present on the sex chromosomes. That means either X chromosome or on the Y chromosome. Now they are of 2 types: X-linked inheritance and Y-linked inheritance. X-linked inheritance, we are getting example hemophilia, color blindness, thalassemia, and Y-linked inheritance is pattern baldness, hypertrichosis. These are the examples. Now, X-linked inheritance, generally we find it in males because males are at a greater risk. Now, why is it so? Because recessive genes on X chromosome. These X-linked inheritance diseases are caused because of the recessive genes of those diseases which are present on the X chromosome. And you know that X chromosomes are dominantly found in females, but male also carry one X-bearing sperm. So, recessive genes means definitely males, because on X chromosome, females will have only the dominant genes. I hope it's clear. So that is why all the X-linked genetic diseases are caused in males, and Y-linked inheritance diseases are definitely found only in males because Y chromosomes are not present in females. So you must learn the examples, and also you must remember why the X-linked inheritance diseases are found only in males. Now, one sum as an example I am showing here. If we cross between a normal mother and a colorblind father. Now, normal mother means definitely XX, and colorblind father we are taking as X degree Y. This degree sign is for showing that the gene or the color blindness gene is present on the particular X chromosome. Okay, we'll show it with the degree sign. So when we are crossing this two, again we are doing a Punnett square and we are segregating the genes. In one of the axes I have segregated X degree Y, and another one I have segregated XX, and we have again multiplied, and we are getting in the offsprings 2 carrier daughters and 2 normal sons, because just now I have said you that only males can carry these genes. So definitely if the females are getting those abnormal genes, they will only be carriers, not the bearers. Okay, so we'll get 2 carrier daughters and 2 normal sons. This is the probability which we are getting. Okay, next, from this chapter from your book, you have to go through the Mendels Law. It is law of dominance, law of segregation, law of independent assortment. Please learn these laws properly, because laws always you have to write exact. And also you go through mutation definition and any 2 examples of mutation from the book. Another thing which you have to learn from the book is this chart. They have given all the characters of the pea plant and their dominant and recessive traits. Please go through this chart, because if you get sums regarding this pea plants for your genetics, if you remember these traits properly, then it will be helpful for you. So students, today up to this. Thank you.